अस्सलाम वालेकुम। आज का चैप्टर है एंजाइम्स। इसमें आता है मेटाबॉलिज्म, द सम ऑफ ऑल biomechanical reactions. Metabolism, or the set of the biochemical reactions that occur in living organisms in order to maintain life, is called metabolism. The term metabolism is derived from the Greek word meaning change. The concept of metabolism was of all given by the Ibn Nafis, who stated that the body and its pathway and its parts always undergoing change. There are two types of metabolism: catabolism and anabolism. Catabolism अब क्या होता है? The process in which large molecules are broken down into simpler molecules by the releasing of energy is called catabolism. Examples इसकी है respiration, ठीक है। उसमें एग्जांपल्स में रिएक्शंस ये है C6H12 O6 और 6O2 goes to 6CO2 प्लस H2O 6H2O और एनर्जी निकल गई। सुक्रोज प्लस वाटर और ग्लूकोज फ्रुक्टोज एटीपी निकल गया। एनाबोलिज्म क्या होता है कि प्रोसेस है जिसमें स्मॉल मॉलिक्यूल्स कंबाइन करके एक लार्ज मॉलिक्यूल बनाते हैं बाय यूटिलाइजिंग ऑफ एनर्जी। इसे एनाबोलिज्म कहते हैं। एग्जांपल इसकी फोटोसिंथेसिस आ जाती है। इसके रिएक्शंस बताने हैं। द कैरेक्टरिस्टिक्स ऑफ ऑल लिविंग थिंग्स सच एज ब्रीथिंग, रिप्रोडक्शन, ग्रोथ एंड रिस्पॉन्स टू स्टिमुलाई are the outcomes of the biochemical reactions taking place inside the body of a living organisms. All these reactions are controlled by a specific protein called enzyme. Enzymes. Enzymes are the biological catalysts which speed up the chemical reactions without alter itself. The term enzymes was first of all used by the German scientist Wilhelm Coney in 1878. Characteristics enzymes के इसकी nature, catalyst, specific for reaction, use in small amounts, specific active center है ये, sensitivity, intracellular enzymes. पहली nature. Enzymes are protein in nature and are secreted by the cells. Catalyst, they act like a catalyst and speed up the chemical reactions. Specific for reactions. Enzymes are specific in the action. They are not specific only for substrate but also for the kind of biochemical reactions. Use in small amount. Enzymes are required in very small amount as compared to the amount of substrate. Specific active center. Enzymes have specific active center called active site which is attached to the substrate. Sensitivity. Enzymes are sensitive to the change of the temperature, pH, etc. These may stop the activity. Intracellular enzymes. Some enzymes are found inside the cells called intracellular enzyme, such as mitochondrial enzyme. Extracellular enzymes. Some enzymes work outside the cells called extracellular enzymes, such as lipase, amylase, pepsin, etc. Remain unaffected. Enzymes are not consumed during the biochemical reactions and can be used again and again. Lower the activation energy. Enzymes do not initiate the reaction but increase the rate of the reaction by lowering the activation energy which is required for substrate. Enzyme inhibitor. Some substance interact with enzymes to prevent it from working is called enzyme inhibitor. For example, poison, drugs, etc. Need effector. Some enzymes work properly when a cofactor is attached. Cofactor is a known protein organic chemical compound that is required for an enzyme as a catalyst. It can be considered helper molecule for enzymes. The main type of cofactor is prosthetic group, coenzymes and activators. Prosthetic groups, it is a type of cofactor that is firmly bound to enzyme and cannot be removed without denaturing is called prosthetic groups. एग्जाम्पल में FMN आ जाएगा और FAD आ जाएगा। उसके बाद है coenzymes, type of cofactor derived from vitamins that is loosely bound to the enzyme and can be readily separated from the enzyme is called coenzyme. Activators, these are the metalines which form temporary attachments with enzymes such as zinc, iron, etc. Activation energy. The energy required to initiate a chemical reaction is called activation energy. All chemical reaction required activation energy. Enzyme lowered activation energy. When a chemical reaction takes place, some amount of energy is required to break chemical bond in a reactant substrate. Such energy is called activation energy. Enzymes lower of the amount of activation energy, thus in the presence of enzyme reaction proceed at a faster rate. Enzymes act in many ways to lower the activation energy. For example, they change the shape of substrate molecule or bring them in a correct orientation. Example, for the reaction अब ये रिएक्शन है, द एक्टिवेशन एनर्जी इज 86 किलोजूल per mole is required without catalyst, and just 1 kg mole energy is required in the presence of enzymes. This is a graph. Mechanism of enzyme. There is a small portion in enzyme that is involved in catalysis. It is in the form of the depression or a pocket on the surface of the enzyme molecule. When an enzyme attached with the substrate, it forms enzyme substrate complex. The active site of enzyme catalyzes the reaction and into product. The enzyme substrate complexes breaks and the enzymes and the products are released. Enzyme substrate मिला के enzyme substrate complex बनेगा और enzyme plus product end पे. There are two models which explain mechanism of enzyme action. Logan key model. This model was presented by the German scientist Emil Fischer in 1894. According to this model, act as a lock and substrate act as a key. Main points: as one specific key can open only a specific lock. In the same manner, the specific enzyme can transfer only specific substrate into products. The enzyme must have the correct geometric shape to fit the substrate. According to this model, the active site has rigid structure. There is no modification in the active site before, during, or after the reaction. Enzymes are generally very specific in their action. They are specific not only for substrate but also for the kind of the reaction. Every enzyme, due to the specific chemical structure, can recognize a particular substrate. Enzyme that catalyze one reaction may not catalyze another. एग्जाम्पल्स इसके कैटलेज, प्रोटीज, एमाइलेज, लाइपेज वगैरह आ जाते हैं. कैटलेज एंजाइम है जो कि कैटलाइज करता है डिकम्पोजिशन ऑफ हाइड्रोजन पेरोक्साइड. इस तरह प्रोटीज ये एक एंजाइम है जो कन्वर्ट करता है प्रोटीन को अमाइनो एसिड्स में. एमाइलेज डाइजेशन ऑफ स्टार्च, एंड लाइपेज कन्वर्ट लिपिड्स इनटू फैटी एसिड एंड ग्लिसरॉल्स.